Will be describing one of the first computational works in finding the most efficient oligos for a treatment of a neuromuscular dystrophy, that is Duchenne Muscular Dystrophy. This is a joint work with the Department of Medical Genetics and the Ingenuity Lab at the University of Alberta. And my name is Hosna. If you have any questions, I would like to answer them at the end, or you can always email me. Duchenne muscular dystrophy is a lethal genetic disease. It starts being diagnosed at the age of 2 to 4. It always happens in boys, and the rate is 1 in every 2,500 or 3,000 boys, and starts showing itself with the weakness of leg muscles. By age of 10, children are wheelchair-bound, and unfortunately, the life expectancy is very low. It's about 25 years. Duchenne muscular dystrophy, or DMD, is caused by mutations, caused by lack of dystrophin protein due to mutations in the DMD gene. A gene is a large concatenation of RNA sequences and can be considered as the program for synthesis of protein. And what you can see over here is animation or a scheme of the DMD gene, and each number over here shows one exon, which is an RNA sequence. And so you can see that the DMD gene actually is very large, has 79 exons that fit together like pieces of a puzzle. On the top, you can see exons of DMD genes from exon 48 to 55. In majority of DMD patients, deletion mutation on any of these exons causes the severity of the disease. On the middle figure, you can see that if exon 52 is deleted, it causes an out-of-frame mutation, which means there is a premature stop codon, and the patient doesn't have any dystrophin protein, and that's why they have severe DMD phenotypes. On the other hand, if we have exon 51 and 52 deleted at the same time, falls into category of in-frame mutation, which causes the patient to show milder phenotypes or sometimes no phenotype at all. The whole point for exon skipping is to transfer the severe DMD type to the milder type by removing some of these exons that cause misfitting of this big puzzle. How do we do that? Let's see some background information, and we can come back to the exon skipping problem. As I mentioned, exons are RNA sequences. RNA sequences are single-stranded, but they don't stay single-stranded. They fold on themselves to form complex three-dimensional structures. If we flatten these structures, we get the secondary structure as the one shown in here. Secondary structure is sometimes presented in two formats. One is the structure you can see, and the second one is an arc diagram. How do we get the arc diagram? We take the two ends of the distinct ends of the RNA sequence, pull them apart, but assuming that the base pairing between the bases don't disrupt, we get arcs that show the base pair representation. The two figures shown in this figure are equivalent. Sometimes the structure we have is more complicated. So we have non-nested base pairings, or what we call as pseudoknot. Here you can see the pseudoknotted base pairs as the green lines. If we show them in the arc diagram, you can see that these arcs are crossing, and that's the hallmark for pseudoknotted structure. Now in this figure, we have part of exon 51. As mentioned, exon are RNA pieces. And how do we do exon skipping? They have figured that if we cover part of the exon, doesn't matter which part, but if the binding between what we have, or we can cover part of this exon, then the exon will be skipped, and we will have a successful exon skipping. How do we do that? We have a small RNA-like molecule to cover any part randomly. For example, you can see that we are covering this unpaired regions of this part of the exon with something that we call oligonucleotide. Oligos for treatment of Duchenne muscular dystrophy are usually synthetic type of RNAs, and the ones that we have focused in this work is morpholino. So the ideal is that this oligo will only bind to this specific exon, and the binding is strong enough so it won't come off in vivo. But the unfortunate thing is that there is no computational framework that gives you how to design these oligos to have the best efficacy. And most studies only involve in vitro testing of oligos, with no guarantee that what they find as the oligo is going to be the optimal one. And if the binding is not as strong, even if you have a strong binding in vitro, it might come off in vivo, and that's one of the main reasons that some of the clinical trials for DMD are failing, in our opinion. So what are the challenges? So the first method, as I mentioned, some of these people are using is exhaustive search. They're looking at the window size, they're trying everything they can. A more complicated way would be to look at the window size, find the unpaired region of the structure, and then aim those to design the sequences, the oligo sequences. But the question is, how long should your window size be? On the right, you see the whole exon 51, which is 233 bases, but we're not considering the surrounding area of the exon. This is the structure you can get. If we consider the surrounding structure, intronic regions, then you can see the structure on the left is completely different from the structure on the right. So if the oligo is designed based on the structure on the right, some unpaired regions on the right are targeted, which may not be unpaired in the real structure, which has a larger window size. And then you don't have a good efficacy. So window size is important. The second way is that what kind of method are you using to create that structure? We know that creating a pseudoknotted structure is an NP-hard problem. So if you're just focusing on the pseudoknot-free structures, only nested structures, you can get the structure on the right side. But if you open up your search space to consider some of the pseudoknotted structures that can form within your prediction program, then you can get a structure similar to the left side with many crossing base pairs. The next question is, most softwares handle single molecules, while in here we have two molecules. We have the exon and we have the oligo. And also, changing the chemistries between oligo and exon is important because they don't have the same type of chemistry. So you have to have a software that considers that as well. While many studies, in vitro studies, consider the oligo to be a small molecule, about 25 to 30 bases, and therefore do not have any structure, as we can see in this slide, oligo can have a 30-base oligo, can have a very complex pseudoknotted structure. In fact, based on our personal communications with Dr. Aoki of Oxford University, the structure of oligo is going to be determinant in leading it to inside the cell to perform a better exon skipping. We have designed one of the first methods, maybe the first, algorithm that considers the interacting structure between exon and oligo. Our program is very time and memory efficient. That's the main problem with most algorithms that consider pseudoknotted structures. Considers large window size, handles pseudoknotted structure, and takes into account that when we're targeting oligos and exons, each one of them may have their original structure, and that this original structure can change based on their interaction. The idea of the algorithm is actually quite simple. We connect oligo and exon with a dummy linker. The complex part is how to assign energy values. And because the loops of the exon sites should be treated differently with from the loops in the oligo side and the loops that are forming between the two. Over here, you can see that this is what our algorithm can predict as the structure of exon 51 when we have 200 bases of flanking sequence. This is the structure we consider for one of these oligos, and our algorithm can use these two structures to predict the interaction between the two. Zooming in, you can see that the structure of exonic sequence shown here in blue has changed to accommodate binding to oligo, and that's what our algorithm can consider. Future direction of our work is that we are testing the efficacy of oligos predicted by our algorithm in vitro and in vivo. We have access to both mouse model and dog model of Duchenne Muscular Dystrophy, and we are hoping that this will be one of the main works for other genetic diseases that can benefit from exon skipping. We have started putting all our data and our algorithm on GitHub. We are also trying to make our framework a bit larger because this is a very primitive view of exon skipping, and there are other factors that contribute to efficacy of this. That's why we are using machine learning. We are using VECA right now to incorporate other affecting factors for exon skipping. And this work was a collaborative work between Ingenuity Lab and Department of Medical Genetics, mostly Dr. Toshio Kota. If you have any question.
A nice talk. I'm just wondering, so to actually confirm the secondary structure prediction, there's another way is using phylogeny derivative structure. So if you just measure the conservation of a crossing species of the same gene, then you actually generate a covariation structure. Have you actually tried that to generate a much more accurate structure?
No, we haven't. You're right in the covariance and using the alignment to find the better structure. But we can see that the genes in different organisms are different and the exons are different. So while the method can be used for different organisms, the same structure is not conserved. So that's why we couldn't do something like that.
Also, there's actually a software in the VNA package called RNA duplex. Have you actually tried RNA duplex measurement just to see how it is?
Yes. So what RNA duplex can do is that it can treat the two sequences as two RNA.